The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elife.elifesciences.org.
Hello, welcome to episode 15 of the eLife podcast, which is produced by The Naked Scientists. I'm Chris Smith. This month, why flies might hold the key to a new generation of fat-fighting treatments, what cannabis does to the connections between nerve cells, and can we cure HIV? Or at least give patients a treatment holiday. But first, influenza. It's naturally an infection of aquatic birds, and it periodically hops into humans, triggering a pandemic. Disease modeller Colin Russell suggests that there are some key things that absolutely need to be done to reduce the threat and increase our chances of controlling flu in future.
In the last 100 years, we've had 5 influenza pandemics in humans. 4 of those pandemics started with viruses that came directly from animals, and one of them happened probably as a product of an unintentional laboratory release. There's a tremendous diversity of influenza viruses that are circulating in animals right now. In chickens, in pigs, horses, basically every animal in which we've ever looked hard enough to find an influenza virus, we find influenza viruses. We're almost certainly going to have another influenza pandemic within the next 10, 20, 30 years. But the question is, which virus is going to cause that? It's important that we figure out which virus is going to cause it, because our only good preparations right now to combat the next pandemic are the development of vaccines and the development of antiviral drugs. And it's particularly the development of vaccines that requires very specific knowledge about the virus that's going to cause the pandemic.
So it's a bit catch-22 because until we know what's coming, it's very hard to defend against it, but we won't know what's coming until it gets here.
Well, that's really the difficulty. And so right now we operate under a very simple model whereby viruses that we observe to cause infections in humans are more likely to cause a pandemic than those viruses that we have not observed to infect humans. However, in 2009, we got a pandemic of an H1N1 virus from pigs in Mexico.
No one saw it coming.
We didn't see it coming. And we want to avoid the repeat of the same situation where we think we know what we should be paying attention to, but in fact, we get blindsided by something else entirely.
So using the power of the connected world we now live in and our better understanding of influenza and how it spreads and circulates, is there a better way to predict what might be around the corner? What are you proposing in this paper?
Well, what we propose is actually sort of a roadmap of things that we need to pursue in order to do a better job of assessing risk. We need further experimental work in the laboratory. What are the attributes that a virus needs to acquire in order to be able to transmit efficiently from humans to humans? Once we have a better experimental handle on figuring out what these attributes are, it would be really good if we didn't have to rely on wet laboratory experiments. They're very time-consuming and they're very expensive. And so if we had a new virus that emerged today and then we had to go into the lab and do a bunch of experiments on it, that would take a lot of time and a lot of money, when we might actually be better spending that time actually responding to containing that virus because we're better able to assess that as a threat.
What are you proposing instead then, that we do this in a computer?
Basically. So ideally, we'll eventually get to a place where we can just get the genetic sequence of that virus, and then from that genetic sequence be able to infer all of the risk characteristics of that virus.
How?
That's where the tricky part comes in, because right now we're a long way from being able to do that. We need a combination of more experimental work, and we need a combination of the development of new computational methods that we can use to infer those characteristics from genetic sequences.
Is that because we're just looking at very large numbers of viruses and marrying up what those viruses did, what the outcomes were, what sorts of pathogenicity, how ill they made people, in other words, were associated with them, and then saying, well, if we look at the new virus that's just emerged, does it have or share any of those particular traits we've seen before, and marry those traits together to make predictions?
Exactly. It's those individual traits that you just started to refer to there, where things like the pathogenicity of the virus, if we can link that pathogenicity back to specific genetic mutations, then we can make inferences about newly emerging viruses. But one of the real difficulties that we face right now is that we don't have a good handle on if mutations in one virus cause the same effect if we saw those same mutations in a different virus.
I can see how that will work very well for viruses that we know exist and that we have some experience of dealing with. But just in the last few years, we've discovered that bats may be carrying a new strain of flu no one had ever heard of. So what about viruses that are unknown?
And they're, of course, the trickiest ones. And so, in addition to this experimental work and the computational tool development to make better inferences from genetic data, we also need better surveillance in animals, and we need better surveillance in humans too. We actually need a more systematic analysis of where we should actually be looking for these viruses to emerge. Most animal influenza surveillance is done in a very ad hoc way. We perceive there to be a problem, and so we go and do surveillance, rather than figuring out where these viruses are most likely to be transmitting and where they have the greatest potential to transmit to humans. And so to that end, we could imagine doing an analysis whereby we look at the distribution of livestock populations, figure out where the greatest density of those populations are, and where humans are most likely to be exposed to those populations. We also need better surveillance in humans, because right now we have good surveillance here in the UK. But for all of the places where we do have good surveillance, there's a whole lot of places where we have effectively no surveillance at all.
Don't tell me, I bet those are the hotspots.
Well, that's the difficulty, right? Because it's in those places where humans have most contact with animals that these viruses are most likely to emerge. And it's the places where humans have most contact with animals that tend to be the poorest and have the least public health infrastructure, and thus the least ability to actually detect when these events are actually happening.
Self-fulfilling prophecy.
In some ways, it's difficult to imagine how it would be any other way in terms of the ability to put emphasis on the detection of emerging viruses when you're at the same time having to deal with a bunch of diseases that don't affect more developed countries.
Colin Russell from Cambridge University. We all know the sensation that comes with feeling full after a good meal, and that most likely contributes to us stopping eating more. But exactly how and where that message is produced and then relayed to the brain wasn't known. Now, working on flies, Yale scientist William Olds has found a group of stretch-sensitive nerve cells that, if you turn them off, makes flies overeat. If you increase the activity in these cells, on the other hand, the insects undereat. And because equivalent nerve cells also exist in humans, targeting this system could be a way to help people to slim down.
I was really interested in metabolism and obesity. And so when I started this, I was just looking at the metabolism of flies. You have all this wiring from the brain to the digestive system, and it's already been established that if you cut any of these wires that connect the gut to the brain, you're going to eat more and you're going to have issues with your feeding control.
And that's even in flies. So you interrupt the nerve connections between a fly brain and its gut, and flies gain weight.
Yes, that is correct.
Gosh, do you know why?
We're still trying to figure that question out. So the hypothesis is that these nerve cells and this communication between the gut and the brain is integral to telling us, hey, stop eating. To test that, we first used some tricks that you can do in flies that allow you to turn on or turn off nerve cells when you want, where you want. So what we did was we inactivated these neurons and activated these neurons, and we just looked at their metabolism, so sugar levels in their blood and their food intake. We found some nerve cells that connect to the posterior part of the fly's gut. So the analogous region would be the colon in humans. And we found that these neurons that are tightly wound around this part of the gut have this profound effect on food intake. If you turn them off, the food intake is 3 to 4-fold higher than controls. And if you turn them on, it's about 1/2 to 1/4.
When you say turn them on, do you mean that you make the cells more active?
Yes.
So there's some
correct.
kind of loop going on here where these cells are informing the brain of something, and if you suppress them, they're sort of releasing the brain, and the brain just says eat.
Precisely.
So what do you think they're actually doing in the fly? Are they there to say to the fly, you're full, stop eating?
That's the current hypothesis I'm working with. In America, we have Thanksgiving, and the whole point is to eat as much as you want, right? Until you get that feeling when you're full. Everybody's experienced with that feeling when you just have 5 or 6 hamburgers and you just feel like you can't eat another bite. You have this feeling of like stretchiness.
5 or 6 hamburgers, Will.
Yeah.
Only 5 or 6.
Well, you know, this is America. We have a terrible obesity problem.
But your conclusion would therefore be that these nerve cells might be stretch receptors. They're detecting how full or distended the guts are, and perhaps they're feeding back on the brain saying, I'm stretching myself to the limit, stop eating.
Precisely.
Are these nerve cells exclusive to the gut, or will you find them doing jobs elsewhere in the animal? I'm thinking therapeutically, if we wanted, and we are able to find these cells existing
Mhm.
in mammals, and we want to target them, for instance, as an anti-obesity treatment, it would be much easier if they were exclusively and selectively present in the intestines and nowhere else in the body.
Mhm. Yeah, so that's a great question. So these are the homologs, the evolutionary conserved versions of these are expressed in multiple regions in mammals. So you have versions of these in your kidneys as well as your lungs. They don't all necessarily play this stretch function that you see in flies. But the nice thing about them being expressed in this intestine region is that the amount of drug that you have to put in is much lower than something in the central nervous system, the brain, for instance. So you have less of a chance of off target effects. This is a very attractive drug target as a result.
To a certain extent, have we not been aware for many years that the gut does appear to be sensitive to stretch? I mean, it A, is logical, and B,
Hmm.
we have got evidence from various lines and various routes that actually if you stretch the gut or distend the gut, signals that appear to be linked to satiety come out. So what does your study add that we didn't really know already?
What we know right now is the stretch is important. You can go back to Homer in the Odyssey. There are multiple passages where they say they go until their stomachs cannot be stretched anymore. So it's obviously clearly something that we've known for a long time. But the question is, who are the players? What is exactly going on? How are these nerve cells able to detect that this stretch is going on? We know they are, but what is giving them the capability to do that? So what my study shows is that these particular stretch receptors are likely the key player in helping the neuron find out when we have eaten too much and that the gut is stretched and you need to stop eating.
William Olds. Receptors for cannabis-like chemicals are present in both the adult and the developing brain. But what's their role? Zsolt Lenki has been using time-lapse video techniques to watch how embryonic nerve cells respond when they're exposed to substances that block or activate cannabis receptors. And his results offer an intriguing explanation for why some cannabis users are at increased risk of developing schizophrenia.
These receptors are sensitive to neurotransmitters and they are also sensitive to cannabis. And we were mapping the localization of these proteins. And we were very surprised that we found a lot of these receptors in very young developing neurons. And this was surprising because these receptors are mostly known as the regulator of synaptic transmission between neuronal cells.
Why were you surprised that there should be so much of the activity of these receptors in a developing brain? Because after all, it's producing lots and lots of nerve cells, and it's producing lots and lots of connections between nerve cells as those cells develop the brain.
We were surprised because the expression of this receptor came very early, when neurons just started to be defined as neuronal cells. They did not have any axons or dendrites. The expression of these receptors was very high during the growth of these neurites, during axons that grow to contact other brain regions. And very surprisingly, when these regions were reached and synapses started to form, these receptors just started to disappear from neurons.
Do you think that that might be because when you've got a developing brain and you want cells to grow a bit, if you let them connect too soon, they may make lots of aberrant local connections and not grow to their target before they start connecting to it? Could it be that that's also part of the issue?
Absolutely. Correct connections are very highly regulated in the brain. In order to have the right number of connections, cannabinoid receptors are working as a brake. We started to think that maybe in developing neurons, they can also have a role, a braking role.
So how can one find out whether or not these endocannabinoid receptors are stopping nerve cells connecting to each other, albeit prematurely?
So in the beginning, we were growing neurons in dishes, and we treated these neurons with pharmacological ligands, molecules, that would either stimulate or block the activation of this receptor. And we have found that molecules that activate the receptor will lead to neurons that grow shorter axons and less dendrites, while blocking this receptor led to neurons that had longer or more developed neurites. Then we wanted to have a closer look at the mechanism of this negative regulatory effect. So we set up video microscopy. So we have young neurons from the embryonic rat brain in a dish. We transform genetically these neurons, so fluorescent molecules will label microtubules and also the cytoskeleton. We put these neurons under a microscope where we film 20 or 30 neurons, typically 1 or 2 hours. And we were very surprised because as soon as we put molecules that stimulate the activation of the receptor, growth cones of the growing axons responded with a very strong contraction.
So on the surfaces of these growth cones, which are the structures that the developing nerve cells are putting out to grow and find a target, ultimately to connect to it, there are receptors for these endocannabinoids. If you stimulate those receptors, this makes the growth cone collapse and retract. Do you understand what's going on in the cell to force that retraction?
Simply by looking at the contracting growth cone, we have seen that F-actin, which is an important cytoskeletal polymer, is redistributed exactly at the places where the contraction takes place.
So putting all this together, what we've got is nerve cells from very early on in development are expressing a receptor for the brain's own cannabis-like chemicals. This receptor is connected to systems that can rearrange the cell's skeleton, and this leads to the forcible retraction of these growth cones. Just extrapolating this, we know that there's an association between cannabis use and psychosis. Do you think that there could be a connection here? Excuse the pun.
The brain is very sensitive to cannabis in the adolescent period. In the adolescent brain, there is a final large-scale remodeling of connections, mostly in regions which are important for cognitive functions. And interestingly, in brain areas which are remodeling in the late adolescent period, you find a lot of cannabis receptors. So overstimulation by heavy cannabis use may lead to large-scale remodeling. These individuals will have less connections, and psychosis is known as a connectivity disease. So these molecular mechanisms that we describe can explain why psychosis will develop in these individuals.
Absolutely fascinating. Joel Lenke, he's at CNRS in Paris. This year, about 2 million people will become infected with HIV. To put that another way, by the time you finish listening to this next interview, a further 20 individuals will have contracted the virus. Currently, this means lifelong treatment with antiviral drugs to control it. These are expensive and they can sometimes have severe side effects. But John Frater from Oxford has found a way to spot patients who might be able to periodically stop their treatment, but without any ill effect.
So we have been interested in trying to find out if it might be possible to cure HIV infection. And one of the big barriers to that has always been that if a patient starts therapy of HIV but then stops therapy, the virus comes back. And it's always been assumed that the virus will come back immediately. And what we found out in a cohort of patients who are quite unusual was that when the therapy was stopped, the virus took a while to come back. And in some cases, up to a year, for a virus that actually can replicate very rapidly and normally can re-emerge in the body within days of drugs being stopped. So the questions we wanted to ask were, how long does it actually take for the virus to come back? Why are these patients different? And is it possible that we could actually predict whether some patients might be in a situation where they could stop therapy for a bit? If you like, go into a period of remission. If you wanted to think of HIV like a cancer, you could have a remission from your HIV. So you could come off therapy and have a period of drug freedom before you had to go back on again.
How did you explore these patients? Do you think they were a very unusual subset, or do you think that they're representative of the broad population of people who have HIV, and we just need to know how to spot people like this in that population?
What is interesting about these patients is they were all diagnosed very early. The history of most HIV infection and the majority of patients that we see in clinic is they're diagnosed often a few years after they were infected, during which time the virus has taken hold, it's spread through the body, and it's sort of laid itself down in a number of different places. With these patients, they were all diagnosed and started on treatment within around 6 months. And actually, most of them were started on treatment within a few weeks of being infected. And because of that short time interval, it looks as though something unusual happened and the virus didn't take hold as well as it might have done otherwise. So when therapy was started, the effect was much greater on the sort of the total viral burden, if you like. And this meant, we think, that when therapy was eventually stopped, most of the patients did rebound reasonably quickly, but in a few of those patients, there was this long delay. And we think it was that very early therapy, which is unusual, which gave them that advantage. And this would then argue for screening for patients early to find out if someone has been infected and get them on therapy very quickly, because that might make a difference to their sort of longer term outcomes.
But obviously, to screen for something, you've got to know what you're looking for. There's got to be some kind of marker. So do you have any feeling as to how you could identify people who fit into that category?
Yes, so the first thing is to identify patients with primary infection. Once they've been diagnosed and started on therapy, the question is then, is there another measure that we can use to say you might be someone who could have a period of remission? This is not yet within any sort of clinical algorithm or protocol, if you like, and everyone has to stay on therapy. But we're thinking about the future. We're looking for sort of tests or biomarkers, is another name for them, which can help us predict patients. And in this work that we've done recently, there was one biomarker, which is a measure of how much viral DNA is hiding within the blood cells. And if you actually quantify that with a very sort of precise assay, that marker was a predictor of when the virus came back, which was a surprise. We always knew that marker was a predictor of how your disease might progress if you became unwell, but we didn't know that it would predict actually whether the virus would wake up or not. And that was sort of the difference that we were just able to discover.
In other words, the interpretation of what you're finding, that the more of that DNA there means more cells are infected, and that person's more likely to have a rapid reinstatement of their virus if you stop treatment?
Yes, so what we found was that the patients who had the most cells containing viral DNA were the ones who rebounded most quickly. On the other side of the coin, the patients who had only a little bit of DNA hidden within their cells behaved slightly differently. Now, some of them also rebounded quite quickly, but a proportion of those ones with low DNA had this very sort of protracted duration of being off therapy without the virus coming back.
Why do you think they have a low risk of the virus coming back? Do you think that just reflects the fact they don't have very much virus lurking in them in the first place, and therefore if there's a chance that it's going to reactivate, if you've got less virus there already, you've got fewer rolls of the dice, so it's less likely to happen and it takes longer before it does?
Yeah, it may be something that simple. I mean, I think one of the caveats to what we've done is that we have taken blood samples and we've only looked in the white blood cells for DNA using a reasonably simple assay. But what we didn't do, we didn't look in other places where the virus can hide, in the lymph node, in the gut, bone marrow, even in the brain. And so when the virus comes back, it could be coming back from any of those places. Our measures are sort of a marker of what might be going on elsewhere in the body. But I think you're right. I think essentially it comes down to sort of the burden of disease that you have on board. And if you can reduce that down to a very low level, then the odds or the chance of the virus coming back are statistically less.
Oxford's John Frater. If two related animals develop in a very similar way and they look almost identical as embryos, even down to the level of individual cells, one might reasonably assume that the genetic mechanisms controlling the process are probably the same in both. Not so, says Lionel Christian, who's been comparing two distantly related species of sea squirt.
My lab studies the development of marine organisms called sea squirts. And the reason why we studied these animals is because they have very few cells that they use to make their heart. So we can study this by looking at every single cell. And we study the genes which are activated in these cells and eventually tell the cells what to do. So in this particular study, we were wondering about how the mechanisms that control the development of the heart are conserved between very distantly related species. because their embryos are almost identical to this. Every cell is organized and different tissues are organized in very similar ways. But these animals have diverged more than 500 million years ago, but they look identical as embryos. And so we were wondering what are the underlying genetic mechanisms that allow this conservation to have the same organization, even though the genetic material is completely different.
So are they using the same genes and controlling them differently, or are they using the same control mechanisms but different genes in order to arrive at this very similar outcome?
Okay, so this is a very good question. We found that to make their heart, they use the same genes at about the same time in the same cells. Okay, so the gene activation patterns remain the same in spite of this great evolutionary distance. What we found is that the mechanisms that control these activities are now the ones that have changed a lot.
Does that mean then that if you were to take the control mechanisms from one species and put them into the other species, they just wouldn't work, despite the fact that the genes are pretty much the same?
Yes, exactly. In other words, one species cannot read the genetic code of the other species and vice versa, even though their own genetic code has evolved to maintain the output the same.
Why do you think this has happened?
Our favorite hypothesis is that every gene is activated by a small group, maybe about 5, of proteins called regulators. And each regulator can activate many different genes, not all the genes, but many different genes. And we think that the cells, they may contain up to 50 regulators. So one way to change in evolution is that if the same gene, instead of being activated by the same 5 regulators every time, draws 5 different regulators from the pool of 50 which are available, such that the gene will still be activated as time goes by and species change, but the exact 5 regulators that control it may change.
Why would that be a beneficial strategy for the organism to adopt?
I can only speculate, but I would argue that this mechanism would buffer against dramatic changes that would end up being lethal. It is not clear that this would be an advantage. What is clear is that it happens. And so you may take it from the angle of it happened because it can happen, because the cells have the potential to change the underlying mechanism and yet keep the outcome the same.
So what do you think the implications of this particular finding are? Because now obviously we have a lot more insights thanks to your work into how this particular subgroup of organisms develop and how evolutionary time has affected the way they control their genes. But what are the broader implications here?
One of them is, what do model systems mean? Because, for example, here we have 2 species. If we look at the way they develop and gene activities, they look the same. And so we could have focused on describing the mechanisms to regulate gene activity in one species and eventually assume that those mechanisms were going to be the same in the other species because the input and outputs are very much the same. But we found this rearrangement of the underlying mechanisms, which now suggests that model species may, for biomedical research, for example, may point to a direction, and some of the mechanisms are likely to be conserved in humans, for example, or in other model organisms, but then the underlying relationships between the parts may have been rearranged a little bit.
Lionel Christian, he's at New York University. That's it for this month. Don't forget that all of the papers we've been discussing are freely available alongside previous editions of this podcast from eLife's website, which is at elifesciences.org/eLife. My name is Chris Smith. This is a Naked Scientist production, and you can find out more about us from our website at nakedscientist.com. I'll be back next month for another look inside eLife. Until then, though, thanks for listening and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elife.elifesciences.org.